All right, hi everyone. Welcome to our workshop today. Today we'll be talking about the Neuron Python package. If you haven't had a chance to review any of the Python coding courses, I recommend doing that. Otherwise, I know it's a busy time of year. Finals are coming up. Still, don't be afraid to just dive in to this code today without, if you don't have a lot of experience with coding or anything like that. Um, because I think the best way to learn how to code is just to get into it. So still dive into this today, even if you haven't had a chance to look through those other coding modules. So go ahead and fill out these two attendance QR codes. I'll give everyone a couple minutes to do that. All right, I'm gonna go ahead and move on. All right. So again, here's our team.
Shit dick. You hear me? Shit dick.
Hello. Okay. So here's our agenda today. We'll be doing so we did our attendance and our pre-assessment. We'll do a little review of the paper just to get a fresh review on that, and then talk a little bit about the Neuron Python package. And then we'll go through an activity just to start to get the familiar with this coding package. Then we'll talk about how we felt with that coding activity, and then move along to the post-assessment as well. So review of the paper. Um, Again, this paper was called A Computational Model of the Escape Response Latency and the Giant Fiber System of the Drosophila melanogaster, which are the fruit flies. So again, we were trying to understand why this escape response latency was increasing with age, and what biophysical properties could be causing this difference. And they were looking at conductance for a lot of this as well. So big picture, we were looking at how these fruit flies were escaping danger, how the circuit changes. Is it a physiological change or maybe it's more anatomical? And then we can also use these computational tools for future targets as well. So if we remember the giant fiber system anatomy, we had these interneurons that descended from the brain to the thorax, and they had these two pathways for jumping and for flying. And again, here we see a more simplified version of the schematic. Um, We have GF represents the giant fiber here. And then we have these different muscles and these different pathways corresponding to those muscles. So for the jump pathway, we're going down the GF to the TTMN to the TTM. And for the flight, we are going from the giant fiber to PSI to DLMN to DLM. And with these, we have different synapses as well, either electrical or chemical. And then the difference that we saw again was in the different latencies. So we saw larger latencies in the old versus the young ones. And we saw that this increasing latency was also related to the gap um, conductance. So we see that there's a higher conductance and lower latency with the young ones versus the old ones had lower conductance and a higher latency, which makes sense. Now, intro to Neuron. How does this relate to the Neuron Python package? How did they use the Neuron Python package to get these results? We'll have an introduction to that today. So first, what is Neuron? What is this Python package used for? We're creating these biophysical models of individual neurons as well as circuits. And how we create these biophysical neurons is based off of measurements of physical properties of neurons that we get from actual data in research experiments. And they simplify these neurons using cylindrical compartments. So these sections to segments. And again, this is all based off of anatomical data. And now there's different types of ways to model neurons. So they had one of the ways that you can do this is with the Hodgkin-Huxley ion channels. Um, so we'll see more about that later. And now we see that they were using gap conductance, and this is how they differentiated between young and old. And the reason why Neuron is helpful is because we can use Python to change these parameters. And it's also very open, which is very related to our theme of this whole program is creating open and reproducible computational neuroscience. So now we have a live coding activity. So if you have your computers nearby, I'll have you scan this QR code to get to this Google Colab notebook. And actually, let me put the link in the chat as well. So I'm going to switch it really quick, but don't worry, I'm coming back. So this is the link to that Google Colab notebook. So go ahead and open that up. And when you open that up, um, first save a copy to your own drive. So by doing that, you'll do file and then save a copy. And then when using Google Colab, um, you'll have to connect to what's called a runtime. So it just does it automatically when you run your first cell, but just so you know that usually it may take a couple seconds, but that's just because it's connecting to the runtime. And then Google Colab has a structure where it has different cells that you can execute. So it's good for prototyping. And so you don't have to like rerun your whole script over and over. You can just do it incrementally with each of these cells. And then in this Google Colab notebook, we have some examples as well as some challenge problems at the end. So hopefully everyone has had a chance to open up that notebook. If not, the link is in the chat. I'm gonna go ahead and switch on over to the notebook. Um, you can do like a reaction in the chat or just say yes or no in the chat. But have you used Google Colab Notebook before? Okay. Nice. All right. Well, perfect. We're all going to be learning today, so that's amazing. That's the whole purpose of this program, is to learn something new. Um, so this helps me just to know where to start with everything. So hopefully have your own notebook open. If you have any issues opening it, let me know in the chat. But what you'll want to do once you have it saved in your own drive is we'll first want to run this first cell. Um, and what's cool about the Google Colab notebooks is that we don't have to download anything physically on our computers. We don't have to install Python into our computers. We don't have to install Jupyter notebooks onto our computers. It's all online, which makes it very user friendly to start with. Um, and you can still do pretty much all the same functionality as a normal Python environment. And you can also have a mixture of text as well as code to help kind of explain what you're doing with these code. So we see, okay, this is our Neuron Python coding. We're going to be getting more familiar with the Neuron Python package. And again, if you haven't saved a copy in your drive, do that. So you'll go up to File, save a copy in Drive, and then you're good to go. So we'll go ahead and run our first cell together. So you'll see the code is in these types of cells. If you get to a notebook, you won't really be able to there'll be no place to run the code. So you're in a code cell when it looks like this. And to run it, you can hit this little play mark, and we see that went pretty fast. And this first um, code right here is you're just printing a statement called hello world. So, that's like the basic first line of code that everyone always runs. So now you're officially a programmer once you run print hello world. So once you've gotten this far, open up your notebook. If you could give me a thumbs up reaction, just so I can make sure we're all on the same track. Okay, I'm gonna continue on, but if you have any issues, just let me know, or don't be afraid to shoot me a message after this workshop. But we're gonna keep on going along. So now hi everyone. So now we'll be going into the Neuron Python package and talking more about that. So we saw earlier how to get it all set up. So now we'll focus on running our code. So you'll want to do a cell first to just get your runtime started and everything connected. And now we'll go into the Neuron Python package. So first we will have to install the Neuron Python package using this command here. What's nice about this, it'll just be installing it on Google Colab, so it won't be doing it locally on your computer or anything like that, so you don't need to worry about that. Next, we will talk more about the Neuron package. So we can see here that we're using this package in this line, and we're importing the specific object here. We'll also be using a um, plotting package as well. So here we just have some standard files in this, which allows us to run our simulation as well. And here we're focusing on creating a neuron. So we're creating a neuron called Soma, and we can see that we're naming it here in this section. Then we are setting the length, which is L, and the diameter of that neuron, which is diam, and we're setting those both to be 20. And then we are inserting HH, which this stands for Hodgkin and Huxley, which is a type of neuron model. So we're just specifying that with this line here. Next, we want to see what happens when we stimulate these neurons. So that is what we'll be doing with these lines here. So we're saying, okay, with our stimulation, we're going to set that equal to this eye clamp, and we're going to be stimulating this soma neuron, and we're going to be doing it at the midpoint. We know it's at the midpoint because for this parameter here, it can be any value between 0 and 1. So 0.5 is right in the middle, so that'll tell us that it's in the midpoint. Then we need to say, okay, um, how long do we want to delay from the start of the recording to stimulating? So this says, okay, 5 milliseconds into the recording, we will do one stimulation. And we'll say that duration of the stimulation lasts 1 millisecond, and the strength of that stimulation is with this parameter here. Then we'll record the neuron and we'll put it in this voltage vector. So we will be acting like we are recording the membrane voltage, and we'll be taking a sample of that membrane voltage at very quick time points so that we can get a good idea of what is going on in this vector, in this neuron. So we're storing those membrane voltages in this vector, and we are recording it on the soma neuron in the midpoint as well. And then with that, we'll also be creating a time vector so that we know at what point in time is this membrane voltage changing. Now we can actually run our simulation, and we'll be running it for a total of 40 milliseconds, and then we can plot the results here. So this is our membrane voltage. We stimulate it at 5 milliseconds, and then we can see what the response looks like. But we know that neurons don't just exist by themselves, they exist with one another in a circuit. So we'll be looking at what a simplified version of that looks like. So again, here we'll be creating two neurons now called cell 1 and cell 2. And what we're doing in this line is we're using a for loop. So if you're not familiar with a for loop is it's saying that for every cell in this list of cells, we're going to be doing these set of commands. And this is very useful when you have a long list of cells and you're going to be doing the same set of commands for each of those cells so that you don't have to type out the code individually, but you can just do it in this loop. So here we're setting the length and the diameter of the cell to both be 20 again, and again using that Hodgkin and Huxley model. Next, we'll be creating a synapse. So this is new because this will be part of the connection since we're having two cells that are connecting. So this synapse will be on the cell 2 because that's what we'll be connecting to. Now this is a single exponential synapse on the midpoint of cell 2. So there are different types of synapses that we can do, but this is the one that we'll be using for this example. And then we also have a time constant, which will also describe the synaptic conductance for the synapse. And then we can also set a reversal potential, and we're doing that to 0. So we'll have no reversal potential because we'll be acting like this is an excitatory neuron. So now we want to connect cell 1 to cell 2. And we'll use this using the function called netcon, which stands for network connection. And again, we'll be connecting it on the midpoint of cell 1 to the second cell. And the second cell will fire when it crosses this threshold. So we set that to be 0. And then you can have a synaptic wait, and we can also set a delay. So this says how long are we going to wait um, to simulate that axonal conduction delay. Then again, we've seen this before. We're going to stimulate that first cell with all the same parameters, except now we're going to see two responses because those two cells are connected to one another. So we're going to create a time vector, our membrane voltage for each of those, and then we'll run our simulation, plot it, and we'll see now that we see two responses, even though we only stimulated one neuron. And that's because they are connected. So you can see now how we can add more and more and more neurons, which we will continue to do to replicate the figures in the paper using the Neuron Python package. Now this is the same plot, but we also added latency, so we can kind of visually see what, we keep on talking about this escape response latency, but now here's a more visual representation of that. In this notebook, I also have some challenge problems for people. If you want to get more familiar with this Python package, I believe the best way to learn how to code is just to get in there and just start coding. Even if you don't have experience, even if you haven't finished the Python models, just try and get in here, solve these questions, and you'll just continue to learn about Python as you go. So we have some practice problems as well as the answers to those problems. And then next, we'll so hopefully everyone felt good about that coding activity. Um, if you didn't, feel free to reach out to anybody on our leadership team. We're here to help you guys and anything that you might need. I'll probably be making another video going more in depth about Neuron, just so people have enough resources to feel comfortable using this Python package, and I'll be giving some more practice problems as well if you feel like you want to do those. And here's just, connecting how this package relates to the paper that we'll be doing, and to also remember that the measurements that we set in our neurons to base them on actual measurements from neurons. So, today we were just using arbitrary values, 2020 for whatever, but in the future we'll be basing it off of these values here. Again, these are the figures that we will be creating soon and what's upcoming. So, continue learning about Python and GitHub if you can. But I would prioritize, going through this notebook, understanding what's in this notebook first. And then we'll be finishing up our module about the Neuron Python package before moving on to the Brian 2 Python package. You'll be getting your group assignments soon so that you can start to work on replicating those figures, but we will be here to help you as well. And then we'll start to see if we see any differences between these two Python packages and work on writing that up in a paper format. So here's our post-assessment. And that is it for today's workshop. If you have any questions, please let me know and have a great rest of your day.